ఎవరైనా సరే స్కూల్లోనే ఉండాలి, చదువుకోవాలి. పిల్లలకి అనేక తెలివితేటలుంటాయి, అవకాశాలు కావాలి. అవకాశాలుంటే మీరు కూడా ఎలాంటి అద్భుతాలు చేయడానికైనా సాధ్యం.
Monohybrid cross. Procedure followed by Mendel is as follows. Mendel selected a variety of plant and from their seeds he grew more plants. Initially, he performed self-pollination to obtain pure breed of the plant. Then, on this pure breed, he performed cross-pollination. He cross-pollinated a pure breed of yellow seed-bearing plant and green seeds-bearing plant. Mendel performed his experiment on 2 pure breeds of peas with yellow and green color. First filial F1 generation. Mendel performed cross-pollination between pure breed of yellow and green peas. After pollination, the plants formed are referred as first filial F1 generation or progeny of the first generation parents. Filial means progeny. All the pea seeds formed in F1 generation are yellow. Second filial F2 generation. He again self-pollinated F1 pea plants and the plants formed as a result of this are called as second filial F2 generation. In F2 generation, 75% yellow seed pea plants are produced and 25% green seed pea plants are produced. Out of this 75%, 25% pea plants are pure breed or homozygous yellow and 50% pea plants are heterozygous yellow. The remaining 25% are green seed producing pure homozygous type. Mendel reasoned that the F1 generation carried the information to produce green seeds even though they did not express this trait. The information for green seeds, which must have come from parent generation plants, had been hidden in F1 generation. He hypothesized that trait for yellow seeds is dominant to the trait for green seeds. To explain his observation, Mendel proposed some assumptions. Assumption 1. Every pea plant has 2 factors which are responsible for producing a particular character or trait. The determining agent responsible for each trait is called a factor. Mendel carefully chose the plants which do not produce a mixed result, which is pure. In our example of yellow and green peas, a pure breed which is parental stage will have both the factors of the same type. A pure breed which is parental, yellow seed producing pea will have both the factors of same type and is denoted by capital Y, capital Y. A pure breed which is parental, green seed producing pea will also have both the factors of same type and is denoted by small y, small y. Capital Y and small y are called homozygous as the factors in them are same. If the combinations is capital Y, small y, then it is called heterozygous. Assumption 2. During reproduction, one factor from each parent is taken to form a new pair in the progeny. Assumption 3. One of these will always dominate the other if mixed together. The trait expressed in F1 generation is called dominant, while the other, which did not express in F1 generation, is called recessive. Assume that capital Y, the one causing yellow color, is dominant factor. That means if capital Y and small y come together, capital Y will dominate. Then the pea seeds will be always yellow in color. From assumption 2, the breed after cross-pollination will have one factor from pure breed yellow, capital Y, and one from the pure breed green, small y. That is, all the peas will have the paired factor capital Y, small y. By the assumption 3, all the peas will be yellow as capital Y factor is dominant. All the pea plants are yellow in F1 generation. The trait expressed in F1 is dominant, not expressed is recessive. This is the law of dominance. Self-pollination in F1 generation. On self-pollinating these peas, one with capital Y small y factor, the new breed have any combinations of capital Y and small y. It can be capital Y, capital Y small y capital Y, or small y. All of them are in equal ratio. F2 generation. So, in this heap, we will get approximately equal number of capital Y, small y, and small y peas. But any pea that has capital Y factor will be yellow. Any pea that has both small y will be green. Since all combinations are equally likely, capital Y will be approximately 25% and is yellow. Capital Y small y will be approximately 25% and is yellow. Small y capital Y will be approximately 25% and is yellow. Small y will be approximately 25% and is green. Some seeds appear yellow in color in F1 generation. When these seeds were sown, some of the plants produce green color seeds. So, we can't determine the internal character based on the external visible character. Phenotype. Thus, in F2 generation, we can clearly observe that 75% are yellow seed producing pea plants and 25% are green ones. This is known as phenotype, externally visible characters, and this ratio is called phenotypic ratio or monohybrid phenotype ratio. It is 3:1. Genotype. Genetically, in 75% yellow seeds producing pea plants, only 25% pea plants produce yellow seeds that are pure breeds, which is capital Y, and are homozygous, that is, to have the same factors for representing a character. Remaining 50% yellow seed producing pea plants are capital Y, small y, heterozygous. The remaining 25% green seed producing pea plants are pure, small y, homozygous type. The constitution of pea plants as shown by representative letters capital Y and small y to show the probable nature of factors is known as genotype. This ratio is known as genotypic ratio or monohybrid genotype ratio. It is 1:2:1. On self-pollinating these peas of F2 generation, we get the following. Case 1. When capital Y peas are self-pollinated, they will produce pure yellow peas, that is capital Y. Case 2. When capital Y small y peas are self-pollinated, they will produce 75% yellow and 25% green peas. Case 3. When small y peas are self-pollinated, they will produce only pure green peas.